Welcome to science class. Today we are going to begin combining what we know about evolution with what we know about genetics. One of the premises of Darwin's hypothesis was that organisms vary and that variation is heritable. But Darwin didn't know to what degree heritability worked on a statistical level, which characteristics are genetic and which are not, and to what extent they are genetic versus environmental. Nobody in the world had any idea until Gregor Mendel. They died only 2 years apart from each other, but Mendel's work never became widely known until the 1900s. But, according to Darwin, it is whole populations of individuals that evolve, because the most favorable traits become more common. Populations are groups of individuals of the same species that live in the same environment and produce fertile offspring. While the variation in each organism exists on the individual level, it's not the case that one day an animal that wasn't a bat gave birth to an animal with webbed hands that could fly. Evolution happens very gradually, and the vast majority of traits arise from the interaction of many genes working together. So you need an entire population whose variation among its individuals produces a much greater variation in the overall genetic makeup of that group. From now on, it will be beneficial to think of the gene pool of the population. A gene pool is all of the genes present in a population, including all of the alleles for each gene. In 1976, Richard Dawkins revolutionized biology with the publication of The Selfish Gene. This book taught biologists that they should look at evolution in a gene-centered way. It is the individual genes that survive generation to generation. Favorable characteristics are caused by differences in gene expression, so it is the genes themselves that are selected for, even if it is done unconsciously by the organisms or by the environment. Genetic evolution, then, is the change in frequency of the alleles over time. This still makes sense in the way we have defined evolution in the past. Black-peppered moths became more common because the allele for black bodies became more common generation after generation. In fact, genetic evolution makes even more sense because now we have something physical, an individual unit of change and heredity, the gene, that we can focus on and keep track of how it changes. Let's get started. So a gene pool is all of the genes present in a population, which means we can track individual alleles. For any population, we can derive their allele frequency, which is the number of times a specific allele occurs in a gene pool, given as a percentage. In the United States, 45% of the population is type O blood, 40% is type A, 11% is type B, and 4% is type AB. But that doesn't tell the whole story, because what I just described are the frequencies of each phenotype. Statistically speaking, half of those people who are type A could be carrying the allele for type O blood, because A is dominant over O. Calculating genotypes is much more complicated than calculating phenotypes, but it does a great job at teaching about gene pools and how dominant alleles mask recessive alleles. In this study, almost 24,000 people in Lagos, Nigeria had their blood typed and their genotype analyzed. In this population, 52.9% were type O, 23.1% were type A, 21.3% were type B, and only 2.7% were type AB. But the overall gene pool looks much different. The O allele accounts for a whopping 73% of the total allele frequency. The A allele makes up 14%, the B allele makes up 13%. There's no AB allele because the phenotype AB comes from inheriting both the A and B allele. The reason type O blood isn't as common as 73% is because it is recessive. You have to inherit 2 copies of it to express it. So while many of the type A blood individuals have the allele for O, it's not included in the phenotypic analysis of the population. You can do this with coin flips. I have my students do this every year. Take 2 pennies and flip them hundreds of times, and keep track of your data. If you pretend heads is dominant, you should see close to a 75%, 25% breakdown of the heads trait being expressed over the tails trait. But the frequency of heads versus tails should be about 50-50. These hidden allele frequencies have important implications that we will discuss in future videos, but we are now going to transition to sources of genetic variation and discuss differences in single gene and polygenic traits. Luckily, we have covered each of these in at least some detail before, so this shouldn't be too tough. The first source for genetic variation is mutations. Remember that a mutation is any change in the DNA sequence, no matter how large or how small. Some mutations affect only a single nucleotide that affects only a single gene, while chromosomal mutations can result in changes of hundreds of genes. And recall that a mutation may be harmless, harmful, or beneficial. There are two crucial things with regards to evolution that you have to understand about mutations. The first is that they occur randomly. While it is statistically possible to calculate how many mutations should accumulate in the gene pool over a period of time, it is impossible to know which genes will mutate or when. As I said in the last video, organisms don't get to decide their own fate. Natural selection does. And whether or not nature will select them is in large part down to the random nature of, does my genetic makeup help me survive in this environment? Normally I don't do this kind of thing, but I want to emphasize what I'm talking about here. What do I mean by the randomness of the selection for survivability of traits? When the dinosaurs went extinct, but the mammals did not go extinct, was that because mammals have a survive an asteroid impact gene and dinosaurs didn't have it? No. It was down to completely unrelated traits and genes and characteristics that mammals happen to have that dinosaurs happened to not have. Okay, back to the video. However, I should also emphasize that survival is not a completely random process. Remember that in many species, members of the opposite sex select mates. That is not random, that is calculated. The other thing to understand about mutations is that mutations don't change the gene pool unless they are inherited. You may get skin cancer in your lifetime, but you cannot pass on that cancer because it does not affect your sex cells. When it comes to unicellular organisms, any genetic variation they have is inherited because they are one cell and they only have one set of DNA. But that's not the case for multicellular organisms. A mutation in a sex cell may cause the offspring to express a trait that the parent does not express, and not for the normal reason that they pass on a recessive allele that was masked, but because the mutation arose only within a sex cell, but involves a gene for, let's just say, hair color. Also important for sexually reproducing organisms is genetic recombination during sexual reproduction. The reason you look different than your parents is not because you are a mutant, but because you are a hybrid. You are 50% mom and 50% dad. If that's the case, then why don't my brother and I look exactly alike? We have the same mom and dad, and each parent passed on 23 chromosomes. Am I adopted? No. My mom's 46 chromosomes came from her parents. She only passed on 23 of them to me, but which 23 get passed on is random by nature. Perhaps I inherited 10 chromosomes from my mother's father and 13 from my mother's mother. But my brother only inherits 5 chromosomes from our mother's mother. There's also our father's parents to contend with. That random shuffling of homologous chromosomes during meiosis alone can produce 8.4 million gene combinations in humans. But crossing over makes it even more complicated. Say my brother and I each inherit the exact same chromosomes from our mother. Each of those 23 chromosomes crossed over with the other 23 chromosomes, and the degree to which they cross over is always different. Neither my brother nor I have any genes that are not present in one of our parents, and we share tons of the same genes, but we are still very different. My sister and I, theoretically, could have inherited 45 identical chromosomes, but we differ in the 46th chromosome. That one difference magnifies a huge number of other traits. My sister and I are way more different than my brother and I are. Gene expression and evolution will be discussed in more detail in a future video because there are many important mechanisms about gene expression to point out. A final source of genetic variation is lateral gene transfer, sometimes called horizontal gene transfer, which has been discussed in 2 of our previous videos. Horizontal gene transfer is a natural process where an organism inherits a section of DNA from an organism that is not its parent. What happens is small packets of DNA called plasmids, which typically only contain a few genes, pass from one cell into another through a vesicle. This is similar to how viruses infect their host, except in this case, the plasmid permanently becomes part of the organism's genome. Horizontal gene transfer has only ever been observed in prokaryotes, and it's fairly common. But it is theoretically possible in eukaryotes, but it would be incredibly rare. Horizontal gene transfer is a kind of mutation, but it has a biological cause rather than an environmental cause. Yes, cells do sometimes cause mutations by making copying errors or during meiosis, but those are random events that are not part of the typical biological process. Along the same lines as horizontal gene transfer is the revelation that after we sequenced the human genome, we learned that around 8% of our DNA came from viruses infecting our ancestors, not from our ancestors' own random genetic variation. This would have to have involved a virus only infecting a reproductive cell, and that DNA getting passed down, then eventually spreading to the entire population. For all humans to have this feature, it tells us that the addition of this viral DNA occurred many millions of years ago, before there were humans. The fact that us and chimpanzees share 98% of our genes means that our common ancestor also had all of those genes. And as we diverged from each other and became different species, we slowly accumulated that 2% difference. Let me repeat that. Any trait or gene that an entire species possesses means that the trait or gene predates the evolution of that species. And if different species share a trait or gene, it means that the trait or gene predates each of those species. As an example, the gene that produces hemoglobin produces a protein that is 287 amino acids long. The amino acid sequence for hemoglobin is 100% identical in humans and chimps, meaning it predates both of our species. This viral DNA we find in our genome must have come about from multiple infections over many millions of years. A virus could not possibly contain, let alone infect, a person with that much DNA. But genes can become duplicated over millions of years. So this length of DNA in our genome, which is viral in origin, may have originally been much shorter than it is today and became duplicated over long periods of time, gradually accumulating up to 8%. Now that we have discussed how the gene pool changes, let's end this video with discussing how that influences the population. We will switch to a more phenotypic perspective rather than genotypic perspective here. First up are single gene traits. If a trait is controlled by only a single gene, then it likely only has 2 or 3 different phenotypes. Mendel had the extraordinary luck of studying traits that were all single gene. Because single gene traits only have one variable controlling them, they are expressed in highly specific ways. Pea plants have flowers that are either purple or white. No blending, no other colors, just that. Pea pods are either yellow or green. That's it. If you graph the phenotypes for single gene traits in a population, you get these nice bar graphs, or you could do a pie chart. Here's a pie chart that breaks down the phenotypes for the blood type study mentioned earlier. This is possible because, again, there is a distinct difference in each phenotype for each individual. But polygenic traits don't work this way. Unlike pea pods, which are either green or yellow, humans don't have either black or blonde hair. And even if you looked at all of the people with blonde hair, there are multiple phenotypes that constitute blonde. This is because polygenic traits are controlled by multiple variables. Most traits are polygenic. Think about it. What can you think of when it comes to people where they are either this or that? There aren't many examples. Blood type is one of them. We tend to say, for example, that people are tall or short, but that's ridiculous. The only way that would work is if you say people taller than average are tall and people who are shorter than average are short. But this would mean that 2 people, one who's a millimeter taller than average and one who's a millimeter shorter than average, would each fall into 2 different categories, even though there's almost no distinguishable difference between the 2 of them. Polygenic traits are manifested along what's called a normal distribution. When you have multiple variables, this is naturally how things organize themselves. Because height is controlled by several genes working together, people come in a range of heights, rather than just 2 different lumps of heights. And the ways in which those genes interact together is almost impossible to predict. Picture we take a bunch of balls and drop them. There's a barrier that will force the balls to either go left or right. Where the balls end up is like a graph of single gene traits. They end up over here or over there. This is a Galton board. There are many dividers, but at each division, the balls can only go either left or right. Once again, it's impossible to know where each individual ball will end up. However, out of that totally random pattern, a normal distribution is always produced. You can think of each individual peg as a variable. There are many variables, so we get a normal distribution. Next time, we are going to continue along the same theme as today and investigate how populations evolve as a result of genetic changes. Thanks for watching.